So I welcome you all for today's session. Very good morning to all of you. So today we are here to continue our live session series. So today's topic is one of the difficult as well as ultra important topic from pathology is necrosis. So we are going to super simplify necrosis today. So let me start. Just a second. Okay. So here you can see my screen. Hello everyone. Hello. Good morning. Yes. So class has just started. Okay, so let me start with necrosis. Today's topic in pathology is necrosis. It's a very important topic, very important topic. So let me explain you. This diagram I have shown you many times. I guess you all can understand this diagram now. So here the cell is in homeostasis. The cell is in homeostasis. Normally from head to toe, all human body is made up of multiple cells and all the cells are in homeostasis. That is, they have a particular structure and they perform their function normally. But whenever any stress acts on the cell, the stress may be physiological or may be pathological. Three things happen one by one. The first thing is cell adaptation. A cell will do certain structural changes inside that in order to cope up the stress. There are 5 types of adaptation. We have already taken a session on that. The second is reversible cell injury. And finally, the irreversible cell injury. Now, if you remove the stress, the adaptations are reversible. If you remove the stress, the reversible cell injury is also reversible. But irreversible cell injury, as the name indicates, even on removing, it's irreversible because irreversible cell injury is actually cell death. So it is the other name given to the cell death. Once the cell is dead, you cannot make it alive again. No, you cannot make it alive again. So that is cell death. Now, there are 2 types of cell death. What are the 2 types of cell death? One is apoptosis. We already have a session on that available on the same channel. Okay. And today we are going to continue the necrosis, the second way of cell death. So in the last session also, I taught you why that cell is having 2 options of dying. One is suicide, one is murder. So ultimately in suicide also, in murder also, the person will be dead. But you know, the mechanism will be different. The mechanism, the suicide is silently. The person will not call everyone. The suicide is silently of a single person. But murder can be of multiple person. And the person will shout, help, 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 and call others for help. So basically, suicide is of a single cell. Necrosis is of multiple cell. That's why a group of cells. That's why we compare apoptosis with suicide and necrosis with murder. Second, in apoptosis, no leakage, no inflammation because it is silent. It is like suicide. And in necrosis, the cell bursts and cytoplasm leaks out everywhere and call everyone. So it is like shouting in the murder. So here the cell is bursting and leakage happens and inflammation happens. That is the basic difference between them. So today my topic of teaching is necrosis. So can I start necrosis? Are you people with me? Can you all give a thumbs up? Can we start necrosis? So apoptosis we have already seen in the last session and now I am teaching you necrosis here. What is necrosis? It is also a type of death, the cell death. Okay. And here always inflammation is present. That is the main feature which differentiates it from apoptosis. So whenever I say cell death, I'm having 2 options, apoptosis and necrosis. So main difference between them is inflammation. In apoptosis, inflammation is absent and in necrosis, it is present. Okay. Now I want to teach you 5 types of necrosis today. I want to teach you ultra important 5 types of necrosis today. Okay. To understand the necrosis properly, easily, you have to understand the morphology of a cell. Can you see a cell? You will say, ma'am, which cell is this? It can be any cell from head to toe. Okay, if I say how many type of cells are present in human body, you will see, ma'am, millions of them. I mean, thousands or millions of them. There are many type of cell. We have brain cell, we have neurons, we have skin cell, we have fat cell, we have RBC, we have WBC, we have platelet. These all are cell. But if I give you any cell in a slide or in a diagram, can you identify which cell is it? Yes, you can identify the type of the cell from which tissue it belongs to based on its 3 details. Number 1, we see the outline of the cell. Can you see the shape of the cell? Different cells have different shape. Different shape means different outline. This is known as architectural detail of the cell. The first thing we are having is architecture. By architecture, I mean the outline. That is the shape of the cell, number 1. Number 2, we see the cytoplasm. Can you see the cytoplasm of the cell? Now, cytoplasm can be eosinophilic, can be basophilic. It can be granular, it can be agranular. It can be scanty, it can be moderate, it can be abundant. So, you know, different cells have different morphology of the cytoplasm. In some, cytoplasm is less, in some it is more. In some it is pink, in some it is blue. In some it is granular, in some it is agranular. In some it contains inclusion body and something additional thing. In some it don't contain. So, cytoplasmic detail of all the cells is different. And finally, we see the nucleus. Can you see the nucleus? The third detail is the nuclear detail. So in some cells, the nucleus is central. In some, it is eccentric. In some, the nucleus is small. In some, it is large. So depending on the details of the three things, we identify various types of cells in human body. Can you see the various types of cells in human body? So can you see some of them are RBC, WBC, various cells. Now see, they all have different shape. They all have different color of the cytoplasm, and they all have different color of the nucleus, different details of the nucleus. So based on the three details, the architectural detail, cytoplasmic detail, and nuclear detail, we can identify a cell. Which type of cell in human body we can identify that. You got this concept? If you got this concept, it's very easy for you to understand the differences between 5 type of necrosis. Let me start with 5 type of necrosis. There are 5 type of necrosis: coagulative necrosis, liquefactive necrosis, caseous necrosis, fat necrosis, and fibrinoid necrosis. Okay, liquefactive necrosis is also known as colligative necrosis. Colligative necrosis. Okay, so we can say triple C double F is the mnemonic. You can learn the 5 type of necrosis. Can we say it once? What are the 5 type of necrosis? There is coagulative, colligative. Colligative is liquefactive, fat, fibrinoid, and caseous, fat, and fibrinoid. These are the 5 type of necrosis. Now basically I want you to understand the definition or the differences between the 5. Now you'll say, ma'am, necrosis means the cell is dying. They all are murders. They are not suicide. They all 5 are murder. Now murder is of 5 type. The first thing, the cell can die because of 2 mechanisms, the suicide and the murder. The suicide is the apoptosis, the murder is the necrosis. But necrosis is of 5 type. I mean, the murder is of 5 type. So you have to understand what is the difference in the 5 type of murders. Okay, I will explain you that in the introduction and the definition of each of them. The second, you must understand the causes in each of them, on which you get many MCQs. You should be able to understand the gross, and most important, you should understand the slide of all 5. That is microscopy. I always insist the student, don't write microscopy, draw it. Microscopy is something you don't have to write. You have to draw it and understand it in the form of the diagram. So based on these 4 headings, we are going to complete the 5 type of necrosis. Can I start? Can I start? Are you with me? Can I start with the first one? First one, can I finish under these headings? And in the end, we will fill this table completely. And we will learn the summary in the form of the table. So let's start with coagulative necrosis. The first one, coagulative. Okay, among the 5, which is the most common? Coagulative necrosis is most common among the 5. So that's the only thing written. Coagulative necrosis is the most common type of necrosis. Okay, say yes, it's most common type. Now what is the thing happening here? See, it's a group of alive cell. Can you see? The cells are live. They are alive. They are live. And this is a group of same cell. They converted into coagulative necrosis. They are dead. So you will say what you can see. Now see the 3 things. Can you see the architecture? Say, ma'am, yes, here also I can see the architecture. Architecture matlab shape, outline. But the same shape or outline I can see here. So I can say the architecture details is persisting. Can I say the architectural details are persisting? Whatever architecture is present in the live cell, the same architecture is present in the coagulative necrotic dead cell. So architecture is maintained. But can you compare the cytoplasm of the 2 cells? The cytoplasm here, the color is different. I mean, cytoplasmic details are lost. Cytoplasmic details are lost. They are not persisting in coagulative necrosis. And can you see the third thing? That is nuclear detail. Here I can see one beautiful nucleus in all the cells. But here some cells, the nucleus is absent. It disappeared, pycnotic. In some, it is karyoraxis. In some, it's small. So, nuclear details are also lost. So, only architecture is maintained, but the cytoplasmic and nuclear details are lost. What I am saying? So, see, this is the group of live cell. It is having the outline, it is having the cytoplasm, it is having the nucleus. Now, as I'm saying that this group is getting converted into coagulative necrosis, I mean this group of cell is dead. I told you necrosis occurs of a group of cell. It is not of a single cell, it is a group of cell. So I'm saying that, now see, here also the outline is same. It's maintained. So in coagulative necrosis, it's such a necrosis where outline is maintained, but cytoplasmic details are lost. Here, whatever the cytoplasmic detail color is there, the color of the cytoplasm will change and the nucleus will also lost. It will be lost, or it will be fragmented, or it will be pycnotic. So cytoplasmic and nuclear details are lost. Only architecture is maintained. How many of you got it? So this is the definition of coagulative necrosis. I am going to teach you 5 types of necrosis. The first one is coagulative. You have to understand the difference between the 5. Okay, the difference between the 5 is based on the 3 properties: architecture of the cell, cytoplasm of the cell, and nuclear details of the cell. You have to see what is maintained and what is lost in each of them. Okay, in coagulative necrosis, I am saying the architecture is maintained, but cytoplasmic and nuclear details are lost. No need to learn, it's visible here. Say yes. The same thing is written. Architectural outline persists, but cytoplasmic and nuclear details are lost. That's why the cell is known as ghost cell or tombstone cell. What do you mean by ghost? Ghost का मतलब होता है भूत. Have you seen a ghost ever? I have never seen, but in the movies I have seen. What is a ghost? Ghost is someone who have only outline. Hana, ghosts don't have the inner details. So can I say the cells here are looking like the ghost cell because they have only outline. They don't have inner detail. No cytoplasmic, no nuclear. They don't have inner detail. Or have you seen a tombstone? What is a tombstone? Okay. What is a tombstone? Tombstone is a stone which is placed over the graveyard. So here also the tombstone is a stone which is having only outline, but stone don't have an inner detail. So can I say the group of the cell converted either into ghost or tombstone? This is the definition. So this is coagulative necrosis. So we always call coagulative necrosis, the cells are ghost cells or tombstone because architecture is there, but other two details are not there. You got it, everyone? Huh? This is the definition. What are the three causes? After definition, we have to see the causes of coagulative necrosis. There are three causes. You know, human body from head to toe, all the organs require blood. What happens if blood is not given to any organ? See, ma'am, the organ will have ischemia and necrosis. Which necrosis? So it is the coagulative necrosis due to ischemia to any organ. If any organ except brain, if any organ in human body do not get blood, the cells of that organ start dying because of coagulative necrosis. But if the brain do not get blood, the cells in the brain also start dying, but coagulative necrosis nahi hota. The coagulative necrosis doesn't occur. Liquefactive necrosis occur in the brain. The second is the injury, thermal injury, I mean the burns. If someone is having, just suppose I have a burn over here, the cells of this portion are dead because of coagulative necrosis and Zanker's degeneration. So these are the three examples of coagulative necrosis. Okay, grossly the organ is pale, firm and slightly swollen. Can you see? It's pale, firm and little bit swollen. That is the gross. Microscopically, this is the microscopy. Now what organ can you see? Listen. So I can see a kidney. See the slide, don't read the text. Microscopy, please don't read the text. See, I will explain you everything in the slide. Can you see the slide? I can see a slide. First thing, identify the organ. I told you coagulative necrosis can occur in any organ except brain. So it cannot be brain. Apart from brain, it can be any organ. So which organ is shown to you? I cannot show you all the organs, but I will show you one. So it is kidney because I can see the glomerulus. Can you see? This is the glomerulus and the tubules. This is the glomerulus and the tubules. Now what I can see in this diagram? In this diagram, I can see this portion is the viable kidney, normal live kidney. And this portion is the dead kidney. It is having the coagulative necrosis. And at the junction of the both, I can see the inflammation. I told you necrosis is never without inflammation. Inflammation must present in necrosis. In all 5 types of necrosis, inflammation must present. Because inflammation is something that is differentiating the apoptosis from the necrosis. So inflammation is the hallmark. Got my point? Okay, got it. So see, now what you can see, now compare the live with the dead. Compare the two sides, the live kidney with the dead kidney. At the junction, there is an inflammation. In the inflammation, I can see these all cells are lymphocytes. It's lymphocytic inflammation. It's okay. But compare the live with the dead. Compare the three things. Compare the outline. Compare the cytoplasm. Compare the nucleus. Now you can see the tubules here. See. It can be PCT, DCT, loop of Hanley, any tubules. I can see the cells in the tubules. I can see their outline. I can see their nucleus. These are the cells. I can see their cytoplasm. Now compare the same tubule over here. You will say, Mama, I can see the same outline. Outline is still there. But if you see the cytoplasm, the color of cytoplasm is changed. If you see the nucleus is also disappeared. It is pycnotic, it is disappeared. So I can see the both side, only one thing is there. The outline is maintained. But cytoplasmic details are lost, nuclear details are lost. That's why the cells are tombstone. The same thing I'm telling you again. Whatever I told you in the definition now, the same thing I'm showing you microscopy. Now it cannot be always kidney. It can be any organ except brain. I can show you spleen. I can show you liver. I can show you lung. I can show you any organ. So one side the organ is normal. The one side the organ will be coagulative necrosis. So here cells have all three details. Architecture, cytoplasmic, nuclear. Here the cells have only architectural details. The other two details are lost. And at the junction there is inflammation. So you should be able to identify the coagulative necrosis in any organ. Whenever the blood supply to that organ is hindered. Say yes. Okay, can we go ahead? Thank you so much for the compliments. Thank you. If you have any doubt, you can write down. Okay, coming on the second type of necrosis. The second type of necrosis is liquefactive, also known as colligative. Rather than reading, understand in the diagram. See See, this is a group of live cell. This is a group of live cell. Now see, here it is coagulative necrosis shown to you, and here it is liquefactive necrosis shown to you. What is the difference? In coagulative necrosis, I can see here architecture is maintained. है ना? We have understood. But architecture is maintained, but cytoplasmic and nuclear details are lost. We can see in the diagram. But what about here? What about in liquefactive necrosis? See, ma'am, here I can see the cell, the live cell. But here I can't even see the outline. No. Dot. No outline. So architecture is lost. Cytoplasmic details is lost. And nuclear details is lost. So basically all three details are lost. है ना? So architecture, cytoplasmic and nuclear. All the three details is lost. In contrast to coagulative necrosis, where at least architecture was maintained, here not even architecture is maintained. Here everything is lost. Say yes if you got it. So if all the three details are lost, ultimately, ban kya raha hai? What is formed? The group of cells get converted into what? Into a gel-like material, into a semi-fluid gel, liquid material. That's why it is known as liquefactive necrosis, because of the hydrolytic enzymes. Okay, so ultimately the group of cell, here the group of cell is known as tombstone or ghost, because it is having the outline. Here the group of cell get converted into a gel. Got my point? So it is converted into a gel. So it is converted into semi-solid, semi-liquid-like material, a gel-like material. That's why known as liquefactive. So what is the difference between coagulative and liquefactive? What is coagulative and liquefactive? So in coagulative necrosis, architectural details persist, but cytoplasmic and nuclear details are lost. So group of cell get converted into tombstone or ghost. And now this is coagulative necrosis. But in liquefactive necrosis, as said, all three details are lost. Architecture is also lost, cytoplasm is also lost, nucleus. The group of cell ultimately get converted into a gel-like material. Since it get converted into gel, that's why known as liquefactive necrosis, also known as coagulative necrosis. Say yes, everyone give me a thumbs up. Thanks a lot. Thanks for the compliment. Okay, so you got it. These are the two till now we have seen. What is the examples? Here we know one example, ischemia of all organ except brain, and here ischemia of brain. If ischemia of brain occurs, brain is not getting the blood. The brain will have liquefactive necrosis. The cells of the brain start dying because of liquefactive necrosis. But if any other organ do not get the blood, the cells of that organ will die because of coagulative necrosis. Apart from this, here we have burns also and Zanker's degeneration also. Okay, so here the two examples. One we already know, ischemia of brain, and one is pyogenic infection of the bacteria. You know, bacteria which form pus. Imagine any organ have a bacteria which form pus. You can understand pus is a liquid, semi-liquid, gel-like material. It is formed because of the liquefactive necrosis. Just suppose I have a pus over here. So here actually the bacteria entered in this tissue, whatever tissue, in this tissue the bacteria have entered, and bacteria started killing the tissue by liquefactive necrosis as semi-liquid pus is formed. So pyogenic infections are there. So learn the two examples here very clearly. Okay, now what is the gross here? In liquefactive necrosis, there is a what is the organ here? What is this organ? You can identify its brain. Intentionally, I have shown you brain so that you can learn that liquefactive necrosis occurs in brain. Ischemia of brain. You can see this is a brain. You can see in the brain there is a cavity, and the cavity contains the liquefied material, a gel-like material. So there is a liquefied center present in a cyst wall. It's a cyst. It's a cavity. It's a cyst. This is the gross. Imagine the gross there. There the gross is this. It's pale, swollen, like that. And see the gross here. It's liquefied. It's not pale. It's liquid, gel-like. Now coming on the microscopy. Don't read the text. See the image. In this image, first identify the organ. It's brain. Let me show you. It's brain. So this side, I'm showing you normal brain. And this side, I'm showing you liquefactive necrosis wala brain. And at the junction, you can see the inflammation. See the inflammation at the junction. The same thing. Normal brain and liquefactive necrosis brain. At the junction, you see the inflammation. Now compare the normal with the necrotic area. In the normal, I can see beautiful neurons are there. Oligodendrocytes are there. These are the brain cells. I can see their outline. I can see their cytoplasm. I can see their nucleus. I can see all three details. This is normal. And in the background, there is stroma, the fibrillary stroma. It's normal. So you can see the cells have beautiful all three structures. They have outline, they have cytoplasmic detail, they have nuclear detail. Now compare what you can see. Say, ma'am, ... Nothing. So architecture, no, I can't see even one cell. In this area, I can't see any cell. No architecture. You can see cytoplasm. No. You can see nuclear. No. Ultimately, it's a group of cell converted into gel. It's the cells which are converted into jelly-like material. It's gel. Can you see it's dot, 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 dot. It's all. And at the junction, I can see the inflammation. Say yes if you got it, everyone. Any confusion? So this is the diagram of liquefactive necrosis in which all three details are lost. So there is a cavity containing the necrotic neutrophil, and it contains the bacteria also if it is pyogenic infection. Okay, so that is the thing. Okay, and there is a inflammation. Now coming on the third type of necrosis, the third type of necrosis is caseous necrosis. Okay, so let me compare the three. We have already seen what is coagulative. We have already seen what is liquefactive. Now it's time to understand the caseous. In all the three necrosis, you can see a group of live cell, a group of live cell, a group of live cell. Now see what it is converting into. Here, the group of live cell get converted into dead cell, group of dead cell. They all are dead. Now they converted into dead cell. But I can see architecture is maintained. Cytoplasmic and nuclear details are lost, and that's why the cell is known as tombstone appearance or ghost appearance. I can see the architecture is maintained. If you can see architecture. Here, all three details are lost. In liquefactive, the group of cell get converted into dead, but I can see only dot, 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 dot. So no architecture, no cytoplasmic, no nuclear details, and a group of cell get converted into a gel-like material. It's a gel-like material. What about the third in the caseous necrosis? Here also, if you see, is the architecture maintained? Say no, ma'am. Here we have cells like this. Here I can see something like this. This architecture is changed. It's not maintained. So architecture is lost. Cytoplasmic details is lost. Nuclear details is lost. All three details lost here also. Now you may be thinking, ma'am, what is the difference between liquefactive necrosis and caseous necrosis in that case? Because in both of them, you are saying all three details are lost. I said yes, but here ultimately it get converted into gel. Here ultimately it converted into granular material. So here, the summary is the granular. Can you see? Granular, dry granular material is formed. Here gel-like material, the group of live cell get converted into gel-like material. All three details are lost. But here also the group of live cell converted into a granular material. All three details are lost. So all three details lost here also, but ultimately what is formed, that is different. So here also all three details are lost. Architectural detail, cytoplasmic detail, and nuclear detail. And ultimately, a granular mass is formed. Granular yellowish dry mass. Have you seen cheese? Have you seen cottage cheese? Do you like cheese? So here the necrotic tissue look like cheese because cheese is yellowish in color. It is dry. It's granular. It's granular in appearance. And that's why since it looked like cheese, it's known as caseous. Caseous is a Greek word. It means cheese. So that's why the name is caseous. So everything have a meaning inside that. Why liquefactive is known as liquefactive? Because ultimately gel is formed. Why caseous is known as caseous? Because ultimately cheese is formed. Cheese matlab granular. वहाँ पे liquid बन रहा था, यहाँ पे granular बन रहा है. So here granular is formed. That's why it's known as caseous. So this is the definition. There are 4 causes. 2 of them are bacterial. 2 of them are fungal. You can learn. है ना? The bacterial is Mycobacterium tuberculosis, which cause TB and syphilis. Okay, and the 2 fungus is Histoplasma and Coccidiomycosis. These are the 2 fungus. Got it? Hannah, these are the 4 causes. Grossly, I told you the organ look, the dead tissue look like cheese. Can you see? This is a lung in which TB is there. This is the granulum of the TB inside a lung. This is the diagram of the lung. I can see the necrotic tissue is dry, number 1. It's granular, number 2. It's soft and yellowish. So it's look like cottage cheese. This is the microscopy. Again in microscopy, first identify the organ. I guess it's a lymph node and you all can see it. It's a lymph node. Okay. You can see this is a normal lymph node and you can see this is a necrotic lymph node, and at the junction I can see the inflammation. So basically the thing is same. It is the eyes who can pick it up. And in all the diagrams I'm showing you the same thing. On one side it's normal, on one side it's necrotic, and at the junction I'm showing you the inflammation. Say yes. Got it? So see the normal lymph node. What you can see in the normal lymph node? I can see the lymphocyte. I can see the follicles. The follicles are made up of lymphocytes. The lymphocytes have architectural details, cytoplasmic detail, nuclear detail. All these cells are beautifully, they are circle, they are small, they have scanty cytoplasm, they have nucleus, they have shapes. Hana? So I can see their details. I can see architectural, cytoplasmic, nuclear. What about here? What about here? Can you see even a single cell? No, I cannot see anything. No architectural detail, no cytoplasmic detail, no nuclear detail. So what I can see? I can see the granular debris. The pink color eosinophilic mass in the form of the granular debris. The granular debris are in the form of the pink eosinophilic mass. And at the junction, there is the inflammation. You will see what's new in that. All necrosis have inflammation at the junction, you told us. But here the inflammation is something unique. So that's why you have to highlight. The inflammation is granulomatous inflammation. which is having granulomas in them. So at the junction, there is granulomatous inflammation, which is having granulomas in them, which contains the giant cells. Can you see? These are the giant cells. These are the giant cells are present here. Everywhere giant cells are there, and the epithelioid cells. So it is a granulomatous inflammation. How many of you got it? Can we go ahead? This is the microscopy. Till now, we have seen 3 necrosis. In the end, we will see the table. We are done with coagulative, we are done with liquefactive, and we are done with caseous. Now it's time to see the 4th necrosis. The 4th necrosis is fat necrosis. As the name indicates, it is necrosis is death of tissue, the fat-rich location in human body. The death of the adipose tissue. You know, fat is made up of adipose tissue. So here, it's the death of the adipose tissue. That is fat necrosis. So where in human body fat is present at 3 locations. Abundant fat is present in 3 locations in human body. So it is the female breast. Number 1. So if there is trauma in the female breast, the death of the female breast fat cells occur by fat necrosis. The second is the pancreas. Pancreas have abundant fat. So if there is pancreatitis, there is death of the cells, fat cells by fat necrosis. And third is mesentery. If there is inflammation, is the mesentery. So female breast, which is having abundant breast, pancreas, pancreas, and mesentery. These are the 3 organs which have abundant fat. Abundant fat matlab abundant adipose tissue. So if there is death of the adipose tissue, it is by fat necrosis. So it occurs only at 3 locations. Breast in trauma, breast in case of trauma, pancreas in case of pancreatitis, and mesentery in case of inflammation. So these are the 3 causes. Grossly, it looks like yellowish deposits. Can you see the yellowish deposits? Yellowish deposits. So grossly, it looks like small yellowish deposits. And sometimes there is soap formation. Calcium soap formation occurs, which is known as saponification. In that case, it is having chalky appearance. Okay, this is the gross. Gross is not very important, although. Coming on microscopy. This is the microscopy. Here, the normal organ is not shown. Normally, in all type of necrosis, till now I taught you, on one side it's normal, on one side it is necrotic. And at the junction, you can see the inflammation. Here it is necrotic. And here it is all inflammation. On this side, the normal organ is missing. I cannot see the normal. Okay, but it's okay. The normal organ can be breast, can be mesentery, can be pancreas, whatever it is. But on the other side, I can see none of the cell is visible. All 3 cells, all 3 details are lost. And it is having cloudy appearance. The word you have to pick is the cloudy. The necrotic tissue is cloudy in appearance. Cloudy matlab badal ke jaisa. You can see it's cloudy in nature. And there is inflammation. Inflammation is not special, it's mixed inflammation. So that is the thing. Coming on the 5th and the last necrosis, fibrinoid necrosis. It occurs only in blood vessel, in the wall of the blood vessel. In the wall of the blood vessel, we are having 3 layers: intima, media, and externa. In the wall of the blood vessel, we are having 3 layers. Can you see? This is intima. Okay, can you see this is complete media? Externa is not shown. Now in the media, I can see some pink color ribbon-like material. A pink color ribbon-like material is present in the media. That is fibrinoid necrosis, occurs in the media of the blood vessel in case of vasculitis or immunological injury to the vessel wall. Okay, so what is this pink color ribbon-like material? It's fibrin-like material. It's not fibrin, but it looks like fibrin. That's why known as fibrinoid necrosis. Mind my words, it's not fibrin, it's fibrin-like. That's why fibrinoid necrosis. So microscopically also, you can see a pink eosinophilic hyaline-like material in the blood vessel wall. That's it. We are done with 5 types of necrosis. How many of you got it? We are done with 5 types of necrosis. We will revise it quickly. What are the 5 types? Can you tell me the 5 types? The first is the coagulative. Then we have liquefactive. Then we have caseous. Then we have fat and fibrinoid. These are the 5 types. Now tell me the definition of all. Who will tell me the definition of all? What is the definition? In coagulative necrosis, if you see, can you see this chart closely? Ultra important master chart. So architecture is maintained, but cytoplasmic and nuclear details are lost. Since architecture is maintained, this is known as ghost cell or tombstone cell. See the second type. In liquefactive, all 3 details are lost. No architecture, no cytoplasm, no nucleus details. Ultimately, the group of cell get converted into a gel-like material. In the third type, caseous, here also all 3 details are lost. No architecture, no cytoplasm, no nucleus. But ultimately, the group of cell get converted into granular debris looking like the cheese. In fat necrosis, it is at fat-rich location. You know the 3 fat-rich location. And fibrinoid necrosis occurs in the vessel wall. These are 2 special type of necrosis. How many of you got it? Give me a thumbs up. If you got the definition, I know it's very useful to understand. Students even don't understand they know the 5 type of necrosis. I guess every medical student knows the 5 type of necrosis, but they don't understand the difference between them. What is the minute difference between them? You have to understand that. Okay, now tell me the causes. Tell me the causes. Here, ischemia of all organ except brain. And here, ischemia of brain. Here, ischemia of all organ except brain in coagulative necrosis. Here, ischemia of brain is liquefactive necrosis along with pyogenic infection. Pyogenic infection is like pus. Pus, है ना? Pus is liquid. Here, 2 bacteria, 2 fungus in caseous necrosis. TB, syphilis, histo, and coccidio. Okay. In fat necrosis, the 3 fat-rich location. The 3 fat-rich location, है ना? Human body, female breast, pancreas, and mesentery. And fibrinoid occurs only in vasculitis of the blood vessel. Gross is not very important. You can skip coming on the microscopy. Rather than reading the microscopy, I never read microscopy in my life. No, I see the diagram. These are the 5 types of necrosis. You must understand. Now, this one is coagulative necrosis. This one is liquefactive necrosis. This one is caseous necrosis. This one is fat necrosis. And this one is fibrinoid necrosis. Okay, first have a look. Okay, now please understand the difference between them in the microscopy. Let's summarize and pack it. Okay, here in coagulative necrosis, on one side I can see normal kidney, on live kidney. On other side I can see the dead kidney, the coagulative necrosis. At the junction I can see the inflammation. Okay, in liquefactive necrosis, one side I can see the normal brain because it occurs in brain. On other side I can see the liquefactive necrosis brain, and at the junction I can see the inflammation. This is the inflammation. Okay, in the third diagram, in caseous necrosis, on one side I can see the normal lymph node. On other side I can see the necrotic lymph node, the caseous necrosis, and at the junction I can see the inflammation. Okay, in the fourth diagram, in fat necrosis, here it is the necrotic tissue and here is the inflammation. On other side the normal diagram is missing, but it's okay. And in the fifth one, it's a blood vessel. Okay, now see the morphology. Here in the kidney, I can see the outline on the viable side also, and I can see the same outline on the dead side also. So architecture persists, but cytoplasmic color is changed, you can see, and nucleus is lost. Nuclear details are lost. So this is coagulative necrosis. In the second one, liquefactive necrosis, here I can see the neurons, the brain cell. I can see they're all 3 details. They have outline, they have cytoplasm, they have nucleus, but here I can see all 3 details are lost, and it is a gel which is formed. It is the gel which is formed. It is gel-like material. Coming on the third one, here I can see the lymph node, lymphocytes. They have all 3 details. They have cytoplasm, nucleus, outline, and here if I see all the 3 details are lost. Okay, and I can see a granular material, eosinophilic material. Here it's cloudy in nature. The only word you have to learn. The fat adipose tissue is cloudy. And here it's a band-like, ribbon-like material in the media, pink color. Give me a thumbs up. We are done with the 5 types of necrosis. Now you'll be able to draw it, understand it, and tell me the important points. So if you're belonging to a second prof university exam, second prof student, and you're going to write your university exam, so necrosis is going to be a big question very frequently. Very frequently you get necrosis and you are asked to describe all 5 necrosis. So if you are asked, I want you to write the detail of all these 4 points in each of them. You will get full marks. If it is of 10 mark now, you will get at least 9 if you are writing all these. Okay, so these are the short questions. But if you belong to competitive exam preparation, like you have already passed your second prof, so you are going to write, appear for NEET PG, FMGE, INICT, PLAB, USMLE. So some MCQs for you. So read the question and tell me the answer. Read the question. Necrosis with cell body retained as ghost cell. In which necrosis it is retained as ghost cell? Is it coagulative necrosis, liquefactive necrosis, caseous necrosis, or none of the above? What is the correct answer? What is the correct answer, people? Yes? What is the correct answer? Necrosis with cell body retained as ghost cell. Ghost cell. What is the correct answer? Ghost cell. Yes? So it is coagulative necrosis. Write down in the chat box. Yes, correct. So ghost cell, because here the outline is maintained, but inner details are lost. Inner details, no inner details, only outline. So it is ghost cell. And if I change the question to tombstone also, still the answer is same. And you all are right. Very correct. Very correct. So correct answer is coagulative necrosis. The next question. All of the following organs undergo coagulative necrosis except. Tell me an organ in which coagulative necrosis never happened. Is it spleen? Is it heart? Is it kidney? Or is it brain? I told you one exception. One exception in which coagulative necrosis never happened. So if in human body ischemia occur of any organ, it leads to coagulative necrosis except brain. Brain never have coagulative necrosis. If ischemia occurs in brain, the cells of the brain start dying by liquefactive necrosis. So correct answer here is brain. Okay. If you want the notes of this session, they are available Can you see the chat box right now? In the chat box, there is one message which is pinned. So there is a link in that. If you click on the link, you will get all the notes of all YouTube live session I have taken live every day. So you will get all the notes. You click on the first file, and in the first file, you will get all the notes one by one. Okay, so all the notes are given. Okay, so we are going to continue these live series. Okay, my next session is on a day after tomorrow, 8th of December. Okay, it's on Friday. So it's on 8th of December. So I'm teaching you a topic of the PSM there. So I will teach you diseases, control, elimination, eradication, along with the iceberg phenomenon, along with the various theories for disease causation. So basically we are going to complete the chapter disease completely in that session. So don't dare to miss it. It's going to be super important and super interesting chapter. So again, but the timing is little bit changed on that day, only on that day. So the timing will be 8 a.m. in the morning. Okay, only on this because I'm having some other commitment at 9:30 onwards. So from 8 onwards, we will start the session only for that Friday. But remaining every MWF, we will be live Monday, Wednesday, Friday, 9:30 a.m. in the morning. So if you have any topic which you want me to cover here in the live session series, don't forget to text that topic on this number. Okay, so this is the contact number. It is only for chat support. So if you have any doubt, whatever I'm teaching you, or if you have any other doubt while you are studying patho, pharma, micro, PSM, okay, and you want to ask me, or if you have any topic in your mind and you want me to take it live on the YouTube, okay, and if the many students are asking for the same topic, okay, so we will schedule that topic. So the contact number is 9321001485. Thank you so much for your patience, time, attention. And here is a QR code given to you. If you scan it, you will connect with me on multiple social media platforms, that is Instagram, Telegram, WhatsApp, for further study material which we are going to provide you for your exam preparation. Okay, so this QR code will divert you to my website. It's MedLive by Dr. Priyanka, and you will get connected with multiple social media platform with me via the website. Thank you so much. Wishing you all the best. Study hard. And if you have anything in your mind, kindly let me know. Okay. Thank you so much for your compliments. Thank you. Thank you so much. Thank you. So we will continue this series, for a long time, and we'll try to cover maximum important topics here. Okay. Okay. So ask your friends, colleagues, batchmates, or any medico which you know and interested to study these subject or these medical topics throughout the globe. Kindly inform them that we are going to conduct it free here live on YouTube. Don't forget to subscribe our YouTube channel and press the bell icon if you want the notification for the sessions. Bye-bye. Wishing you all the best.